Recently, I was part of a musical and I had to learn a dance. The dance was taught in sections and repeated many times to gain accuracy. Each time, the director said, "This is for your brain." Now, he's right. Learning any new motor task, or motor learning, takes time. And as we age, the time it takes to learn and master tasks increases. Unfortunately, 40 million people suffer from motor disorders, and it can be impossible to ever learn a new motor task. Think of a child having trouble learning how to walk. The root cause of any motor disease remains unknown, but we know that there's some disconnect in the brain on how to properly orchestrate movements. There are no cures, and current treatment options can often worsen symptoms. Motor learning is a very complex process where brain cells, called neurons, communicate with each other to determine which movements to make. Learning starts by understanding how we move, controlled by the DMS, shown in pink on the slide. We put one foot in front of the other to stay balanced, and as our body understands the sequence, the DLS, shown in blue on the slide, takes control, making the process automatic. Now you're not thinking about how you walk, you just do. Motor learning in mice can happen on the rotarod task, shown on the slide. Initially, mice fall off quickly, but by day 5, with repetition and training, they learn to stay on the rod and they don't fall off. Movements are controlled by dopamine, a neurotransmitter that was released from one neuron that sends instruction to the next neuron. That next neuron is instructed to make cyclic AMP, a small chemical that carries information throughout the brain. We wanted to see if the pink and blue regions use cyclic AMP to coordinate the transition from untrained to learned behaviors. My hypothesis is that cyclic AMP regulates motor learning by dopamine instruction by decreasing the amount made in the blue region after learning. We use a powerful microscope to visualize these neurons in either the pink or blue regions, and I look for cyclic AMP changes in these regions in either untrained mice or mice that have successfully learned the rotarod task. We found that by dopamine instruction, the blue region makes less cyclic AMP in untrained mice, but in learned mice, it makes more. This is a new finding indicating that between the DMS and the DLS, the cyclic AMP production is different, further showing for the first time that dopamine instruction of cyclic AMP in these two regions is different. This is not the full story, however. My work fits into a giant puzzle promising for the field of motor function that improves therapies that increases cyclic AMP in the DLS, so that whether you're dancing through life or you need to learn to walk again, cyclic AMP may be the key to mastery.